Hello everybody and welcome back to a new instalment of Teach Me in 10, the video series brought to you by Technology Networks, where we ask scientists to describe their research area in a grand total of 10 minutes. My name's Molly Campbell and I'm a science writer and editor for Technology Networks, and I'm delighted to say that our guest for this instalment of Teach Me in 10 is Pedro Oliveira, who earned his PhD in biotechnology from the Technical University of Lisbon in 2010. He also conducted his postdoctoral work at the Technical University of Lisbon and later at the Pasteur Institute in Paris. Since 2016, he holds a senior scientist position at the School of Medicine at Mount Sinai in New York. His current scientific interests centre on the bioinformatics and comparative genomics or epigenomics of bacteria, particularly in the interplay between methylation, gene flux, and chromosome organisation. So welcome, Pedro. We are really excited to have you here for a Teach Me in 10 instalment. Do you think that you are up for the challenge?
I hope so. Thank you for the opportunity.
No problem. So without further ado, the timer is set and you have 10 minutes to teach us about your research area.
Thank you very much, Molly, for this opportunity to speak a little bit about my work on microbial epigenomics and on certain future trends in the field. So we know that in the age of post-genomics, the majority of research has focused essentially on decoding the sequences of the 4 canonical DNA bases and also making sense of its meaning. So we know as well that on top of this information, there is an extra layer of epigenetic data encoded as covalent modifications to these bases. So these modified bases effectively expand the DNA alphabet beyond 4 nucleotides in a process mediated by what we know to be DNA methyltransferases. In humans, for example, the methylation at the 5th position of cytosine is known to be an important epigenetic mark that can affect embryonic development, differentiation, among others. So bacterial genomes are also methylated. We have here in this slide examples of methylation at 4MC, 6MA as well. But these methylations, with just a few exceptions, for example, defense against DNA evasion or involvement in DNA mismatch repair or cell cycle control and some evidence on transcription regulation, there is overall little is known on the functions of the multiple DNA methyltransferases that are out there and their corresponding epigenomes. So my current research focuses on better understanding bacterial epigenomes, as well as their diversity and functional roles using 3rd-generation long-read sequencing technologies. As an example that I show here, we have recently found or performed the first comprehensive DNA methylome analysis of a human pathogen, Clostridioides difficile, using 3rd-generation sequencing and comparative epigenomics. We found a methyltransferase whose inactivation was able to impact sporulation, which is a key step to its transmission to a human host, as well as biofilm formation and colonization. And here on the left, you can see an overall depiction of the epigenome of C. difficile, as well as the complete landscape of target methylation sites that we detected using such technologies. So, okay, the great part of methodological development for DNA methylation detection has been dedicated to characterizing 5MC with sensitive restriction enzymes, or by using affinity enrichment of methylated DNA fragments using specific antibodies, or eventually using chemical conversion of 5MCs using sodium bisulfite. So, owing to the different forms of methylation that bacteria contains, the characterization of their methylomes requires alternative detection methods beyond these that I just described. On the other hand, we know that 2nd-generation sequencing technologies have so far offered vast improvements over Sanger sequencing And their limitations, especially their short read lengths, make them poorly suited for certain biological problems, including assembly and determination of complex genomic regions, or gene isoform detection or methylation detection. So to overcome such hurdles, one must consider a technology that is able to provide at least an order of magnitude increase in read length. That's where long read sequencing technologies have made the difference. So here in the slide, we see, for example, on the top part of it, smart sequencing, which can simultaneously report nucleotide sequencing and the 3 major types of DNA methylation in bacteria, although at different sensitivities due to signal-to-noise ratio specific to each modification type. So smart sequencing relies on a sequencing by synthesis approach. A DNA polymerase is bound within a tiny observation chamber called a zero-mode waveguide, and uses a strand from the native sequencing library as a template for the read, and starts to incorporate fluorescently labeled nucleotides as they diffuse into this tiny chamber. So each incorporated nucleotide is briefly immobilized at the polymerase active site and emits a fluorescent pulse in the corresponding color channel. And specifically, in smart sequencing, the time interval between the pulses that signal the incorporation events can describe the polymerase kinetics as it translocates along the DNA template. And this is key to determine the epigenetic status of that base. So a second option that we see on the bottom of the slide is nanopore sequencing, which has been under active development for the last few decades. And recent progress has led to the release of first commercially available sequencing platforms. So the underlying technology builds upon variations in ionic current induced by the passage of different nucleotides through genetically engineered protein nanopores. And although the vast majority of research applications to date have indeed focused on DNA sequencing, this ionic current has been shown to differ between canonical bases and covalently modified nucleotides, which also enables the technology to detect chemical DNA modifications. So what are the key challenges and future directions in the field? So I'll just highlight a few. So here on top, I highlight a question that is quite important, is how can we highlight methylation sites that play important regulatory roles? So in this case, genomic regions with a significant high density of methylation sites should be targeted for site-directed mutagenesis or gene editing in order to really gauge the impact of each methylation. And as alternative, one can think about an alternative strategy using an orthogonal approach to deduce functional relevance based on the degree of conservation of orthologous methylation motifs across multiple genomes. Another challenge that I might think about is how can we specifically detect DNA methylation marks de novo? This is not shown in this slide, but it's quite an important point that should be stressed out. So this results from the fact that existing detection methods were either trained for detecting a specific type of DNA methylation from one or a few specific sequence contexts, or allow for a more general detection without effectively differentiating between different forms of DNA methylation or identifying the exact modified position. So we are in the need of de novo detection designs that can be used to detect additional forms of DNA methylation, of DNA damage, and possibly other forms of RNA modifications. One future direction that I would also like to pinpoint regards phase variable methyltransferases. So phase variation is the random and reversible switching of gene expression, which allows a population of organisms to generate a variety of phenotypic variants. And these variants can be better equipped to colonize certain niches. So these phase variable genes often contain a number of well-defined DNA sequence features, such as DNA repeats, and their phase variation results in differential DNA methylation throughout the genome, which leads to the variable expression of multiple genes via epigenetic mechanisms. So these systems are called phase variants. And a thorough understanding of these phase variable methyltransferases and the phase variants they control is required for a better understanding of bacterial pathogenesis, for example, or to the development of new vaccines and treatments. Finally, I would like to point out that one possible future research avenue is to look at the thermodynamic effect of DNA methylation and its ability to induce conformational changes to a bacterial chromosome, and vice versa. So the generation of methylation-induced non-beta topologies is likely to take place at higher methylation densities and should provide key insight on how structural changes can alter the repertoire of genes exposed to the cellular transcriptional machinery. So in this case, we can use techniques such as circular dichroism or chromatin conformation capture, which can be used to elucidate the effects of bacterial DNA methylation on DNA conformation and consequently on gene expression. So on top of this, it would also be interesting to test the extent to which non-canonical or non-bDNA conformations contribute to the occurrence of non-methylated sites, particularly for those cases that cannot be explained by competition with other proteins. So I guess I would stop here. I thank you, Molly, and Technology Networks for this great opportunity.
Thank you so much, Pedro, for your Teach Me in 10. You've shared some really fascinating insights.
Thank you very much.
Thank you.
Very much. And,
so, to our listeners, if you would like to learn more about Pedro's research, we will include links to his research page and relevant publications in the description box below. And we will be back soon with some more Teach Me in 10s next week. Thank you and goodbye.